a new topic of cell size that we're going to talk about today and also tomorrow. I did want to say a few more words about what we did before. So first thing is just to connect with what's coming to you next week by Amir. I gave you a very simplified picture of the Lenski experiment. Actually, I want to kind of zoom out for a second and talk about the kind of level of description we're working at. So today we're going to talk about single cells, right? How single cells control their cell size, the mechanism, phenomenological, maybe a bit on the molecular size. Okay, so it's all about single cells. Okay, and next week it's going to be all about interactions between cells, because ecology is about how different species interact with each other and so on. But I want to point out that what we discussed in the last couple of days about evolution and population dynamics was in a sense in between, because we had no direct interactions between cells, right? But they were competing, say, for resources through the, say, in the Moran process. Every time a cell divides, you kick another cell out. So that's another kind of form of interaction through the competition for the limiting resources, the space, okay? But I think next week you will see more direct interactions between cells in the models of ecology. And I want to point out that even though in the calculations we did, this was the only interaction between cells, right? That a cell divides and I kick out another cell in the Moran process and fixation and all that. In reality, life is often more complicated, and this is just one example that I pulled from I guess 2 years ago by Lenski, saying that actually, and it's known also before this paper, that if you look at the Lenski strains, okay, so you take them from the freezer, let's say you take the E. coli cells from the culture from, I don't know, 1999, you will not find, in the picture we described in evolution, there is always a single species, right? We spoke about this SSWM regime, the strong selection, weak mutation regime, where you have these genetic sweeps, and after each sweep, another mutant takes over the population. The population is always uniform, right? Maybe except for these very short periods of time where you have two competing strains, it's always one strain of E. coli that overtakes the population. And so the population is very uniform genetically. But in reality, it's not the case. It took some time to know this. How, like, let's say you want to know what's the form, what's the distribution of E. coli mutants and species in the population. What would you do? You'd open the freezer, take out the culture, and do what? What's the key word? Sequencing, exactly. So this wasn't around when Lenski started it in 1988, but now it's very easy and conventional. So people did that in the last decade. They sequenced the hell out of the Lenski strains and learned a lot of things. And one of the things they learned is that there is a whole ecosystem. What you think of as kind of a uniform soup of one species of E. coli is actually one dominant strain. It is true to a first approximation. So what I told you is relevant. But then you have, at a lower abundance, a lot of coexisting species. So when you have coexistence of species, it leads to these questions of ecology that Amir is going to describe next week. So this is one example of a paper by Lenski where they see how a new species of or a new mutant of E. coli evolves that has cross-feeding interactions with another one, meaning maybe you have the main species and it digests some nutrient and spits out another food waste, and then another mutant of E. coli learns to feed on the waste of the main dominant species. That's the cross-feeding they're talking about here. Okay, so there is ecology also in the Lenski strain, and what we did was kind of a first approximation of neglecting these ecological interactions and between the different subspecies of E. coli. One more comment about the sort of philosophy of what we're trying to do. you saw yesterday we did a model that wasn't able to explain the fitness trajectories in the Lenski experiment, right? The models where the genes don't interact with each other was unsuccessful at giving the right power law trajectories you see in the experiments. But it still teaches us something, because then it teaches us we need something else. Maybe that something is interaction between genes. Maybe it's something else, but you need to improve the model. So often the purpose of a model is to tell you what could be. It's not that the model has to fit every detail of reality, and it can be useful even if it fails. So keep that in mind. There is a famous quote, all models are wrong, some are useful, by a famous statistician called Box. This is actually a very good paper that has a lot of other stuff in it. So it's a good read about different approaches to science and so on. I actually checked yesterday, and in that paper, this is often the quote that's used, but in this paper, the quote is just all models are wrong. They don't have the second part. And there is another, I don't know why, yeah, it's here. There is another quote from another very good paper, The Strategy of Model Building in Population Biology. It sounds specific, but actually they talk here, the lesson they give here is very, very general to modeling for all fields. And I've used it when I taught a course on mathematical modeling at Harvard in the past. It's by Richard Levins. And basically in this paper, he takes one example, actually related to ecology, and studies it from using very different models. Okay, so he uses different types of models, very different from each other. One using a graphical approach, one using an algebraic approach, and so on and so forth. And then he finds what is common to all of the modeling approaches. And then he says, therefore, we attempt to treat the same problem with several alternative models, each with different simplifications, but with a common biological assumptions. Then, if these models, despite their different assumptions, lead to similar results, we have what we can call a robust theorem, which is relatively free of the details of the models. Hence, our truth is the intersection of independent lies. Okay, so one kind of small example that we saw this week was somebody asked me when we did like this happened to be on the board, but it's perfect. It's exactly what I need now. We did the Moran process where the population was exactly fixed. Okay? And we calculated the fixation probability in that model and found it to be linear in the fitness. But somebody asked, but how do you know that that's true, say, for the serial dilution, which is what Lenski does, where your cells grow and then they're diluted and grow and they're diluted. And my answer was actually, if you calculate fixation probability within this model or within this model or within other models, the first one where this was done was probably the Wright-Fisher model, a very popular model to study evolution that I didn't discuss this week. The results are identical, up to maybe factor 2, not very important, but qualitatively, they're very, very, very similar between these different models. So you kind of know, okay, that's a robust result. Fixation is proportional to the fitness advantage. So that's something that you usually want to do. You don't want to kind of have a model with 15 parameters and declare victory. You want to have a simplified model and then maybe try some different approach and see that the results are consistent. Okay, so that was kind of a general comment about modeling. Okay, so going back, we're discussing size control. We spoke about cell size being constrained. We spoke a little bit about this FTSZ protein, multiple replication forks. We ruled out the possibility that cells are timing their divisions because we realized that would lead to a runaway, because we realized that this equation is really a random walk in disguise, right? If you take the logarithm of size, it's a more natural variable to work with because cells grow exponentially at the single cell level. And then we get this problem of the random walk, diffusion. And that poses a problem here because it means we need feedback. And we did this simple simulation showing that by adding feedback, we can control size. So now we're discussing what we're going to do today is talk about coarse-grained models for size control that will have this feedback built into them. And then we're going to try to use experimental data to find out which of these coarse-grained models is the relevant one. And from there, we can ask what are the molecular mechanism. And I am going to talk a lot about this question of coupling to DNA replication. And we discussed 3 different classes of model. One where division is completely independent of DNA replication. You accumulate this FTSZ protein from birth until it's sufficient level, and then you start constricting and you divide. Nothing to do with DNA replication. We saw this more complex data suggesting that really maybe size is controlled not for division, but for DNA replication. And I told you that this was actually consistent with a conclusion that was reached more indirectly already long time ago. And I told you that I'll show you the raw data that I kind of sketched on the board. So this is experimental data from 1958, where the x-axis is the growth rate, and the y-axis is the logarithm of cell size. And you can see a very nice quantitative relation. That size is exponential in the growth rate, and the slope here is precisely 60 minutes. Okay, so that's one observation. And the second observation, again, this is experimental data from a decade later, showing that the time from starting a duplication of DNA until cell division is also 60 minutes. This is only true in fast growth E. coli. In slow growth E. coli, it's no longer true. But over this range of doubling times from 20 minutes to 60 minutes, then this is exactly 60 minutes. And this is exactly the regime of multiple replication forks. This is what's called fast growth E. coli. Any questions? Very good. So, I asked Ilya Schechter, I asked Donachie. So Donachie connected these 2 experiments to reach this conclusion. And I asked him, like, how did you feel about taking this experiment on Salmonella and combining it with the experiments on E. coli? And he told me, well, the biggest difference between the 2 is the name. Okay, so Salmonella is a close relative of E. coli. And for this physiological purpose, they're close enough that you can basically treat them as one species. Of course, Salmonella is pathogenic, so for us, it will make a difference if you're infected by Salmonella or by E. coli. Yeah, but good observation. Other questions? Okay, so let's see. Okay, and this was this graphical representation of the 3 classes of model. You sort of should be able to understand vaguely what this means, but I haven't explained mathematically what it means. So Li is initiation. Okay, so this is birth, division, initiation, and in the some round, and initiation in the next round. So when you have an arrow, it means initiation is affecting, say, cell division, or birth is affecting cell division. Okay, so you kind of have an intuitive feeling maybe of what this means, but I haven't quantified what each arrow here means mathematically, and we will have that by the end of today. Okay, so for now it's a scheme, but in fact it's part of a powerful machinery called causal inference that I'm going to mainly talk about tomorrow. Okay? All right. So now we're kind of going to build the tools we need to do that. So ultimately, the goal by the end of tomorrow is that we will have tools to distinguish between these 3 different models using fluctuations. Again, the theme, I guess 2 themes for this week are stochasticity, that plays a big role, and learning from fluctuation, that came also from Luria Delbruck, and you'll see it again here. Okay, so these are the 2 themes. And so now what we're going to do is to develop a coarse-grained model for the size control that has feedback in it, and we saw that we need this feedback on size. And the idea would be in the spirit of the Langevin equation. Who here has seen Langevin equations before? Okay, almost all of you. That's great. So it makes it puts you in a good position to have intuition for this problem. So, in a sense, the simplest Langevin equation is something like this. dx to dt is equal to noise. Okay, no feedback, but just noise. What kind of process is described by this? What is this? Somebody? random walk, diffusion, right? So if I'm just, my position is driven on, say, 1D or 2D or 3D by noise, then I'm just going to do a random walk. And we know that I can write, this is the Langevin equation, but I can write the Fokker-Planck equation. And the Fokker-Planck equation that corresponds to this Langevin equation is what? What do we call it? Diffusion equation, right? So dp to dt equals d times Laplacian of p. Okay? So this is one example, but then Langevin equations are more powerful and more general because they can have something like dx to dt equals a function of x encoding for some feedback, and this can be done in 1 or 2 or 3 dimensions, plus some noise. Okay? And in that case, we can also write a Fokker-Planck equation, and there is a well-known machinery for doing that. And again, if you're interested, you can, early on in my book, I describe Langevin Fokker-Planck equations and a lot of applications for them in fields from finance, like Black-Scholes, to problems in hydrology and quantum optics. And in that spirit, we want to write cell division as some function of cell birth plus a noise, right? So the main insight of Langevin was to say, let's say I look at a Brownian particle, okay, like the ones in the Piran experiment that we discussed yesterday. We have some small particles suspended in a liquid, and it gets collisions from the water molecules. And these collisions do two things. They give rise to a deterministic drag force, right? If I kind of drop this, the air drag will give me a deterministic force that is going to be, say, at low Reynolds number proportional to the velocity and viscosity and so on. But it's also going to give rise to the fluctuations, right? When a particle is suspended in the liquid and moves randomly, it's these random kicks with the same water molecules. So it's one mechanism in the case of the original context of Langevin that leads to the deterministic force, the drag, and to the fluctuations. But splitting it in this way to a deterministic force and the noise is very helpful. That's the kind of key insight of Langevin, to split it into a deterministic component and a stochastic component. Okay, very powerful idea that led to very general results. Okay, so we want to do something similar and write the division as a function of cell birth. This is what the cell is trying to do. So this function f is what the cell is attempting to do if there was no stochasticity. Okay? For example, maybe a cell wants to be born at some size. If it's born at a size v birth, maybe it wants to divide at twice that size. Okay? Maybe it wants to get to a critical size. Okay, so that would be a different strategy. Okay? And then to that, you add noise. And we're going to talk about what's a more realistic form of the noise. Okay? So that's the idea. And now let's talk about examples. So if you're trying to double your size, that's not going to work. Why is that not going to work? That's exactly the timer case we discussed yesterday. Right? If growth is exponential, growing for a constant time is exactly doubling your size, if you time it correctly. So we saw that because of the noise, if you double and divide, fluctuations build up over time. This would lead, log of size will do a random walk in this case. Is this clear? Okay. If this function is a constant, then this is, you're basically trying to get to a critical size. So this is sometimes called a sizer, and this limit is called a timer. Okay, so these are two examples. Okay. And if this function, for example, f of vb is vb plus a constant, what does that mean? You're born a given size, and you're dividing when you added enough volume before your division, before the next division. So this is called an adder because you're adding a constant amount from birth to division. Okay, but these are just three examples of this function that can be completely arbitrary. Okay, so stop me if anything is unclear. Now, we can rely on the biological fact that we saw yesterday, that the size distributions were very narrow, right? The coefficient of variation of the size distribution at birth or division was about 10%, if you recall the data from yesterday. So if I have this f of VB, okay, this is what's characterizing my model. This is VB, and this is f of VB, and this is some function. Now, a model that has an entire function as a fitting parameter is going to be a problematic model to fit. There is too much freedom, right? But what comes to our help is that the size at birth, say in a given condition, let's say your volume was this 1 micron cubed or something like that, but the spread of it was only 10%. That's what we saw in the experimental data. So if we have something that's concentrated in a very limited regime, what can we do to this function to help? Something you all know, I'm sure. I have a function, but I'm only interested in analyzing it in a very small, narrow regime. So what tool is helpful for us in that case? Taylor expansions, exactly. So we can Taylor expand f of VB around the typical. I can write it as approximately the average size, okay, at birth, plus the derivative at the average size times VB minus V naught. So I can take it to the first order. I can take the linear expansion of this function, and that would be a good enough approximation for our purposes. Okay, so that's what we're going to do. And if we do that, then only the derivative will matter, okay, for the size control. The first term will determine the absolute size, okay? But you can measure things in units of the average cell size, and all the distributions and their shapes and the correlations that we're going to discuss today will be only dictated by the derivative of this function at the average size. And a convenient parameterization for this is to call this derivative 2 times 1 minus alpha. Okay? So now we've boiled down an entire function in, being my fitting parameter, okay, an infinite number of parameters, to a single parameter alpha. Okay? So alpha is the control parameter in the model. And so you can see in this timer case, the derivative is 2. And if the derivative is 2, then alpha is 0. So this case of the timer, alpha is 0. In the case of a sizer, my derivative is 0, so alpha is 1. So now we have alpha going from 0 to 1 in a continuous manner, interpolating our timer and the sizer limits. And it's a continuous spectrum, and actually we're not limited to this. Alpha can be larger than 1 as well. What does alpha larger than 1 correspond to in the language of feedback? Like if alpha equals to 1, if alpha equals to 0 is no feedback, and alpha equals to 1 is like getting to a critical size, then what is alpha being 1.5? What is it? It's over-correcting, right? Like you don't just get to the feedback, you have too much feedback. So we'll see that. Okay, so too much feedback is also not good, right? Like if you have a restoring force, if every time you're here you go too far to the left, then it's also a problem. That's essentially what you'll get for too large values of alpha. And we'll see this, okay? And for this other case, the derivative is 1, so alpha is half. So in a sense, this model, in a quantitative sense, is halfway in between these two other models. So in the literature from the last decade, people often talk about just these 3 cases as if they're the only possibilities, but that's not true. There is a continuous spectrum of models connecting them, and there is nothing special about these 3 models a priori. Okay? So now we need to talk about, I want to talk about 2 things. I'm going to start with this. We need to add noise to the model. And again, in the spirit of the quote by Levins, that you want to study different models and make sure that the conclusions are robust, then you could have just added Gaussian noise to the size, and it would have been fine. All the calculations I'm going to do today pretty much will be the same. Okay? But, yeah, go ahead.
Yes, sorry. Now you mentioned that we need to add noise, but when you do this expansion, it mentions a small noise. I get confused.
Okay, it is confusing because I've done this. I've used the empirical fact the distributions are narrow to justify why I can Taylor expand. So now I will do a calculation, and at the end, to be self-consistent, I need to make sure that the distribution that I will get is indeed narrow. And it will be. And that would justify retroactively why we could take the Taylor expansion. So it's often like that in physics, right? Like you make a, you make an assumption, you do the relevant approximations, and then at the very end, you need to make sure to go back and check that you're indeed consistent, and it will be. We will find that the distributions are very narrow. For alpha, we know that alpha equals to 0 is not going to give us that because it's going to give us a broad distribution. We saw that yesterday. But for alpha between 0 and 2, this is what we'll find, we will be consistent. That if the noise we add is small, the distributions will not diverge and will stay small. So that's consistent. Other questions? OK, so we want to now add noise. And like I said, we could add noise to size and it'll be fine. But the more realistic type of noise, and we'll see why that's in better agreement with the experimental data as well, is to assume that the noise is added to the generation times. Okay, so I'm trying to implement some, I know my size, I'm trying to divide at a critical size or at some size, but then I have maybe half a minute delay or half a minute earlier because of some type of fluctuation. So we can add the noise to the generation times. Okay? Adding noise to the generation times is not an additive noise for the size, right? Because growth is exponential, then the size at division is the size at birth e to the lambda t, where t is my generation time. Now, if t has the time I was hoping for, this is my attempt time, and to that I add noise, okay? So you can write this, vb e to the lambda ta is what I was trying to achieve. So this is exactly f of vb, but now it's multiplied by e to the lambda tn. So you can see that if I have, say, Gaussian noise added to my generation times, for the purpose of size, it's a multiplicative noise. I'm getting multiplied by a noise, so the distribution of this is actually, it's a log normal distribution, the distribution of exponential of a Gaussian variable. So the details of the noise will be different. It'll lead to different distributions of size and time. But again, for the purpose of correlations that are going to be the focus of today, you get the same results in both models. If you have size additive noise, or now you see why I will call this time additive noise multiplicative noise for the purpose of size because of this property. OK, so we know how to add noise to the model. And I'm going to kind of focus the calculations on this type of noise. You can try for yourself what would happen if you had Gaussian noise in size. It's actually easier calculations. And for this reason, I'm going to focus now my solutions on this particular family of functions. Okay? F of VB equal to V naught to the alpha. V naught is the average cell size at birth, times VB to the 1 minus alpha. NB is newborn, okay, so it's the same. VB to the 1 minus alpha. So I choose a power law function of VB. Why? You will see in the second. Okay? But I want to make sure that this alpha here is consistent with my previous definition of alpha, because before I defined alpha as the derivative equals 2 times 1 minus alpha. Right? So let's make sure that this is consistent. If you take this power law function and you take the derivative at VB, you will get 2 times 1 minus alpha, V naught to the alpha, VB to the minus alpha. So indeed, you get 2 times 1 minus alpha. So the definition of alpha here in this function is consistent with the previous definition of alpha. Okay? So now, what is the trick we're going to do? We're going to solve the model for this particular family of power law functions. And why does that help us? Because we said that any function that you give me, the only thing that matters is its derivative at the typical newborn size because of this Taylor expansion, right? So if I have all these power law functions, like this and like this, if you give me any function, I can find which of these families has the same derivative as this one, meaning they need to have the same alpha. And if I solve the model with this power law function with the same alpha, I've essentially made sure the two functions have the same derivatives, so it's going to be the relevant one for us. Let me try to explain it on an example, and maybe it'll be clearer. Okay? Let's say I want to solve the model for the case f of VB equals VB plus a constant. This is what we called an adder, and it's going to be actually a very relevant, biologically relevant solution. It's easier for me to solve the model for this time additive noise for the case of a function that is twice times square root of V naught times VB. Okay? Which alpha does this correspond to? Half. Which alpha does this correspond to? Also half. What does this mean? It means that if I draw these functions, this one is tangent to this one at the typical newborn size. So for all the range that we're interested in, these two are essentially indistinguishable. If you simulate the model with this function or with this function, you're going to get the same statistics, the same correlations, the same distribution, up to higher orders that are negligible. Okay? So I can choose to solve the model with this function, and essentially all the predictions I will make will be applicable also to this function. And that's a general thing. Like if you give me any function, I just need to find what value of alpha I have, and I can choose this model with this alpha. So that's why I want to kind of work on these power law functions. And now you'll see why I want to do that. Okay, so yes? Is the same as VB. Yes. Exactly. So VB is birth, VD is division. Okay. So let's see why we want to have this, why I chose this function. Okay, so to summarize, we now have VD equals f of VB plus some noise. And for this function, we have 2 V naught to the alpha VB to the 1 minus alpha. And we have time equals to this attempt time plus some noise. This is the model. Okay? And now it's convenient to work with the logarithm of cell size. So I can call xn the log, let's say, to the base 2 of VB divided by the average size V naught. Okay? And if we now look at xn plus 1, okay, then what do we need to do? We need to take the logarithm of VD over 2, because we have symmetric division. That's the next size, right? Divided by V naught. And now we just need to plug in VD, right, into this. And after some simple algebra, we will find that xn plus 1 equals 1 minus alpha times x. Maybe we'll do it slowly. Okay, so let's do it. So this is the logarithm of size at the next generation. And now I'm plotting. So this is log to the base 2. And I'm plugging in f, so I get 2 v naught to the alpha vb to the 1 minus alpha over 2v naught. And we saw before that by adding the noise to the time, what does that do to the size of division? It's multiplicative. So it came as e to the lambda, right, times this Tn. So now you can see why I chose this power law family of functions. Because when I take the log, everything becomes, the 1 minus alpha is going to drop in front, and I will get log to the base 2 of Vb. So I will get a linear relation between xn plus 1 and xn, and the noise will become additive to the log of sizes. So everything kind of falls into place. Okay, so let's continue. So we find that xn plus 1 equals, the important thing is this, the log to the base 2 of Vb1 minus alpha becomes 1 minus alpha log to the base 2 of Vb over V naught, right? Sorry, let's clean up the V naught. So I can write V naught to the alpha divided by V naught to the 1 minus alpha. It had to be that way so that the dimensions would make sense. Can you see, or should I lower this? Okay. Okay. Let's do that. So you can see that the 2 cancels. Vb over V naught to the 1 minus alpha, when I take the log, becomes 1 minus alpha xn. Okay? And then all we're left with is log to the base 2 of e to the lambda Tn. Okay? Which essentially becomes, what is it? It's up to a constant, just some Gaussian noise, right? Because Tn was Gaussian. And once I take the log, this exponential becomes just linear in Tn. So we get this very simple equation that I'm going to talk about for a few minutes to give you some intuition, and then we're going to solve and find the correlations and the distributions and so on and so forth. Okay? So this is what we found. Okay? Just to be consistent with the slides, I'll keep the lambda also here. Okay? So that's the equation we have. And first of all, just to kind of connect with yesterday, if alpha was 0, then what is this equation? Random walk, and we saw it yesterday. No surprise, it just connects things we did. We saw that alpha equals 0 is the case with no feedback, which is just a timer case where I give my cells a clock. Yesterday, we saw that that leads to a random walk, right? And indeed, that's what we see now. So that's just a consistency check. Now we see that what role does alpha play in this game? When you look at this equation, when alpha is larger than, not 0, then what kind of effect does it have? What is it? Well, deterministic, yes, but what kind of thing does it do for the size? Control, feedback, right? Like we saw yesterday that in the code, when we add a feedback on size, when it's like the size in the next generation is the size minus a 1% times the size, that sort of feedback was enough to control things. So we kind of expect intuitively that this is going to have the effect of feedback. Okay? And indeed, if you simulate this for alpha equals 0, by the way, simulating this is 3 lines of code, right? It's super easy to simulate this. You just need to write that size at the next, or log size, this xn plus 1, 1 minus alpha xn plus noise, and you just need to generate Gaussian random variables. So it's really like 3 lines of code, and you will see that for alpha equals 0, you get a runaway, while for alpha equals to half, you get fluctuations that are confined. They don't run away. This is experimental data. Okay? It's not a proof of anything. We'll learn how to do it quantitatively shortly, but there is hope, okay, that maybe alpha equals to some value captures the statistics of the data properly. Now, before I do the calculations, I also want to give you a bit of intuition. So I spoke over breakfast with some students about OU process. Ornstein-Uhlenbeck process is another example of a Langevin equation. If you just had dx to dt equals noise, we discussed it, that's just a random walk, it's diffusion. One of the simplest Langevin equations that is very useful and appears in all fields of science, of physics, is this one. Dx to dt equals to minus a restoring force times x plus noise. So what does that correspond to? It corresponds to a Brownian particle in a harmonic potential. Right? Harmonic potentials mean you're connected by a spring, and that's exactly this linear restoring force you have here. Okay? So in that case, if I have a restoring force of any value, I will get a confined behavior. This is a confining potential, and I will converge to a distribution. Can anybody guess what kind of distribution will converge to here? Gaussian. It has to be Gaussian. Why? Because of Weizmann. Right? The potential is quadratic, goes as x squared. So Weizmann tells us that the stationary solution must be exponential in the potential. Exponential of x squared is a Gaussian. Okay? So one example of that is the Maxwell distribution of velocities. If you take particles and you ask what is the distribution of velocity, it's e to the minus v squared for exactly this reason. Okay, so this is intuition. In this case, things are a bit more subtle because it's a discrete version. So for the continuous version, we know, like I told you, can always go from a Langevin to a Fokker-Planck equation and solve the Fokker-Planck. Here we cannot do this for this discrete version. So things are a bit harder. And in fact, the math can be quite tricky, and I'm not going to go into these mathematical subtleties. We're going to solve things approximately for the top equation, and that's still going to be pretty easy. Okay, so we're going to solve it approximately, and we'll see that qualitatively it behaves similarly. This alpha has the effect of a restoring force, but we have the danger of overcorrecting. If alpha is too large, then it's also going to fail, and we're not going to get a confined distribution. So what I'll show you now is that we get confined distribution as long as alpha is in the range 0 to 2. Okay? And I'll also show you that biology points to a particular value of alpha equals to half. The correlations and statistics of the actual data often are consistent with this particular value of alpha. So that's what we're going to do now. We're going to solve the model, find the distribution it leads to, find the correlations it leads to, and then use them to analyze data. Any questions? Okay. So there is one paper that I can refer you to that makes an approximate mapping to a Fokker-Planck equation and uses that to analyze the model. But yes, I guess maybe what you're referring to is close to what we did in this paper here, where we kind of write a self-consistent equation for the stationary distribution, and it's kind of an integral equation, and we managed to solve it. But I won't go into these subtleties today. Yes?
Two questions.
Sure.
The first one is about the lens penetration, or say the noise equation you're considering there. Where are you adding the noise? I mean, it's somewhat typical is noise, but I don't see where the E lands at the end.
Okay, so let me go through this again. So the idea is this. I want to assume that cells have an attempt time. Okay? What is their attempt time? It's simply 1 over lambda log of VD over, or log of f of VB over VB. This is where they want to divide. So this is how long they are trying to grow for. And to that time, I'm going to assume there is a noise, Gaussian noise that's added. Does that answer your question? Okay. Second question?
And the question is, can you length pass this
Again.
Can you
Sure. Please. You want to take it? You want to pass this? Okay. Any other questions? Pen. You want? Okay. So let's solve the model, and the first thing we're going to. So now I think. Okay, I'm going to quote the result, and then we're going to prove them, just to kind of give us some motivation of where we're going to do. I'm going to define Pearson correlation coefficients. Who here has heard of Pearson correlation coefficients? Okay, so about half of you. So I'll define, it's a very natural way to measure correlations between two variables. And what we're going to prove is that for this model, if I look at a cell and its daughter cell, the correlation is 1 minus alpha. Okay? And then it's very easy to extend it. In a cell and it's a granddaughter cell, it's going to be, the correlation is going to be 1 minus alpha squared. Okay? So cells n generations apart, the correlation will be 1 minus alpha to the n. And I'm going to compute the correlation between size at birth and the time. So a cell that is born at some size, how long is it going to grow for? And it turns out that the smaller cells want to grow for longer. So it's always a negative correlation, and the magnitude is minus square root of alpha over 2. Okay? And all of this will be true only as long as alpha is between 0 and 2, because we'll prove that this is the range at which you get a stationary size distribution. So we're going to prove all these things. And we're also going to prove, okay, and this is an example of experimental data showing you that the correlation of size at birth and division is about a half. And the correlation between size and time is about a minus a half. And these examples suggest that alpha for E. coli is a half, because it agrees with these predictions for a value of alpha equals to a half. So that's what we're going to do next. And we'll also show, this is actually experimental data, and physicists like to collapse distribution because it often means you understand something, right? So here I'm showing you collapse of two distributions that a priori didn't have anything to do with each other. One is the size distribution, and the other is the generation time distribution. I'm taking E. coli, imagine the videos that I showed you yesterday, and I'm measuring the distribution of cell size at birth, and I'm measuring the distribution of how long it took them to grow. And what I'm showing you here is T over T average minus 1. So I've kind of centered the time distribution around 0 and normalized it according to the average time. And I'm taking, instead of just the size distribution, I'm taking the distribution of log to the base 2 of the size. And we will see why these two distributions are both within the model we just defined for a particular value of alpha equals to half are predicted to collapse, to be both Gaussian and collapse on top of each other. And that's similar to what you see in this experimental data. Okay? So that will, now we're going to spend probably the rest of today doing these calculations. And maybe that should convince you that this is a reasonable model where cells are adding a constant volume from birth to division. Okay? Because that's the model for alpha equals to half. It's this adder model where you're adding a constant volume from birth to division. I don't think that's what happens in E. coli. So what we're going to discuss tomorrow is how you can get all of these results in a different model that is also coupled to DNA replication. So the fact that this model is able to predict all these things is great, but you can never prove a model, right? A model you can disprove. But here we'll find that this model makes all these good predictions that work out for the correlations and distribution, but it doesn't mean that this is how it's working biologically. And we'll show tomorrow, we actually already saw evidence of this coupling to DNA replication with this critical size for DNA replication. So that's where we're kind of going to go with this. Okay, so let's solve the model. And the first thing, actually, I think I'm going to stick with a board for the most part. So let's take this equation. And I want to ask you a subtle question. Let's look at the variable xn. It's a random variable. and let's look at the variable xn plus 1. What is the relation, like, what is the mean of xn? It's 0 because we've centered it around the typical newborn size. Now, it has some variance that we don't know yet. Okay? What is the relation between the variance of xn and the variance of xn plus 1? Not quite. It's simpler than that. So you want them to be the same. That's the right answer. Because think about it. How do you do variance? You take your size at birth for this cell, for the next cell, for the next cell, and you take the variance of that statistics. What is xn plus 1? It's the same list of, very long list of numbers, just shifted by one value. So it's the same numbers you're working with. It must be the same. So we know that xn and xn plus 1, it's not only true for the variance, it's true for any moments. They have exactly the same distribution. They must have. But they're correlated. They're not independent variables. So we'll talk about their correlations as well. Okay, but now we're talking about their distribution. So if the variance of xn plus 1 and the variance of xn is the same, let's call it sigma v, sigma volume squared. And I can write an equation immediately that sigma v squared, that's the variance of the left-hand side, is equal to the variance of the right-hand side. Now, this noise is by assumption independent of xn. By the way, it's not independent of xn plus 1. They will be correlated, right? Because this noise is driving xn plus 1. Okay? But it is independent of xn. So for these independent variables, I can sum up the variance. What is the variance of this guy? You said it. 1 minus alpha squared times sigma v squared. We had this earlier this week, so remember that for the variance you have to square the coefficient. This is important. So we get 1 minus alpha squared sigma v squared plus the noise here that I'm going to call, let me make sure that I'm consistent. I'm going to call it sigma n over tau squared. Okay? So the model has one parameter associated with the size control, that's alpha, and it has another parameter, which is the magnitude of the noise that I add. Clearly, the noise that I add will determine how broad my size distributions will be, right? Because that's what's making them broad. For the correlations, the nice thing is, you already saw it in the previous slide, the noise magnitude drops out. So the noise magnitude drops out, and the correlations only depend on a single parameter alpha, and that's a useful property, because we're not sensitive to these details. But for the distributions, the noise must enter. By the way, I should emphasize now, how many different noises do we have in the model? 1, and that's why the time distributions and the size distributions must be connected to each other. That's why we have these relations between these time distributions and size distributions. They're both driven by one form of noise. It's not two independent forms of noise for each of them. That's why we have this connection in the model. So it leads to another prediction for this collapse of the distributions. OK, so now we've essentially solved it. This is an input to the model, right? This is something we know how much noise. This is the time additive noise we've added. This is its magnitude. So now this is the only unknown, and we found it. So we can find that sigma v squared is equal to sigma n over tau squared, which is dimensionless, times 1 over 1 minus alpha squared. And we can write this as alpha times 2 minus alpha. Okay? So now you can already see where the model would lead to an inconsistency. If alpha is outside the range 0 to 2, we'll get a negative value, nonsensical because this is positive. It means there is no, what did I assume in this? I assumed that there is a stationary solution, right? Because I assumed that xn plus 1 has the same statistics as xn. I've assumed I've reached a stationary solution. Now we can see it cannot exist for alpha outside the range 0 to 2. Okay, so we've learned that stability only for alpha between 0 and 2. And we can also see where will we get the tightest size distribution. What do you need to do? You need to minimize 1 over alpha 2 minus alpha. You can do it in various ways. It's very easy. And what is the kind of minimal width of the size distribution? What's your guess? Anybody has a guess for what you'll get? Alpha equals to 1. That makes sense for several reasons. First of all, it's smack in the middle of the stability range, so it makes sense that that's where your tightest distribution. Second of all, it's a sizer. Sizer, you're controlling the critical size. It makes sense that that would give rise to the tightest possible distribution. Right? Okay, and if you don't believe me, just take this function and take its derivative, equate to 0, and you'll get alpha equals to 1. Okay. Third comment, this is the distribution. Okay, I can do it this way. Look at this equation and tell me, now let's say I don't want just the variance, I want the entire distribution. Okay? What kind of, what variable would you guess for xn in order to solve this? What distribution of xn? Any guesses? Gaussian. If you guess a Gaussian with this variance for xn, then this is a Gaussian with mean 0 and some variance we just calculated. This is a Gaussian. Gaussian plus Gaussian, you all know it's also Gaussian. So this will also turn out to be Gaussian with the same variance. So we know that the distribution of xn is Gaussian. If the distribution of xn is Gaussian, what is our prediction for the size distribution? Remember that x is the logarithm. So what's the size distribution? If the logarithm of something is a Gaussian, then what is the variable itself? Log normal. Okay, so we predict a log normal size distribution. Okay. So this is how it looks like for different values of alpha and some given noise. You will get some distribution that is not Gaussian. It's skewed to the right because it's a log normal distribution. This is what we just found, that the noise looks like this with this alpha 2 minus alpha and the noise magnitude. And by doing a change of variables, again, take it as an exercise, you can find the form of this log normal distribution. So it's e to the this x squared divided by size. Okay, this is what makes it right skewed. Not too important for us. The fact that it's log normal and not Gaussian is not a big deal. Okay, so we're done with the size distribution. Now we can talk about the time distribution. And it's very similar. Like, once you get the gist of it, it's kind of very similar mathematics. The time is this constant. Okay, how do we find the time? It's written right here. We need to take 1 over lambda log of f of vb over vb and add to it some noise. Okay? But f of vb is this power law, right? It's like this 2 v naught to the alpha, vb to the 1 minus alpha. Okay? And you can see that it'll be simply, okay, let's just do it. It's easier than explaining. Okay, so the time is equal to 1 over lambda log of 2 v naught to the alpha, vb to the 1 minus alpha, divided by vb, plus this Gaussian noise. Plus a Gaussian, let's call it psi, plus psi. So now this thing here is simply 1 over lambda log 2. That's simply the average doubling time tau. Okay, so in all of these models, to a first approximation, you grow for the doubling time. The rest is just small corrections due to the size feedback. But the dominant effect is this tau. This comes from the 2. And the rest is 1 over lambda. And then we get log of VB. VB, let's write it as minus 1 over lambda log VB over V naught. And this is to the power of alpha, so we get minus alpha log VB over V naught. And that's exactly the variable xn. So we see that time is a constant minus alpha over lambda xn. It's linear in our xn, plus some noise. And now we're in a kind of, this is basically the equations that's written here. Constant. Okay, this is minus alpha times our variable x plus some noise. So now if I want to find the variance of the time distribution, it's very easy. What do I need to do? I need to recognize that the noise is independent of our variable xn. We've already used it before. So if the noise is independent, I can add up the variances. The variance of xn we've already found. Okay? So I can write that, eraser. Okay, I want to keep this. I'm going to keep this one. So I'll just write it here. So I can write that the variance of time is equal. This constant doesn't contribute to the variance because it's just a constant. So I get alpha over lambda squared times sigma v squared. Okay, sorry, there is another subtlety. This is not exactly our xn. Why not? It's not very important, but it'll lead to typos later on. So let's make sure we get it right. It's because of x was defined log to the base 2, and this is log to the natural base. Okay, so all you need to do is write here times log to the base log of 2 squared. Okay, and now it's correct. So this accounts for this part. Okay, so let's write almost xn. And then we need to add this time additive noise, this sigma n squared. So when you put in now the formula that we have here for sigma v squared into the noise for the time, so the important is not like this simple algebra, but the logic, right? What is independent of what and what you're adding. So this one we know already, so I can plug it in. Okay? And when I do that, I find that, first of all, what kind of distribution is the generation time distribution? Gaussian. That was easy for the time because it was a Gaussian. The distribution of the variable xn was Gaussian, plus the noise, which is Gaussian by assumption. And when you work out, again, you take this formula and plug it in and add this, you get that the variance, or the standard deviation, goes as square root of 2 divided by 2 minus alpha. So for the range alpha between 0 and 2, what kind of dependence do we have? For the size, it was minimal variance at alpha equals to 1. Here, where will we get the minimal variance? Opposite. Opposite, right? So the minimal variance is alpha approaching 0. Why does that make sense? It's not discrete. Like the size distribution diverges in that case. But what happens to the time distribution in that case? It's a timer. So timers are terrible at controlling size, but for controlling time, they're the best. So the time distribution, nothing pathological happens as alpha goes to 0. It becomes narrower and narrower as alpha approaches 0. Okay? So we found the size distribution, and we found the time distribution. Okay? And the next thing I want to do is talk about the correlations. Okay? And for that, I'm going to define the concept of a Pearson correlation coefficient. Okay, so maybe I'll leave these two formulas. And so a Pearson correlation coefficient, if I have two random variables x and y, is defined as the expectation value of x minus x average times y minus y average divided by the standard deviation of x times the standard deviation of y. Okay, this is called Pearson correlation coefficient. By the way, you remember Pearson from yesterday. This was the problem of the random walk. That was his, he invented it. Okay, same guy. What's good about them? They're insensitive if we shift x by a constant, because when I shift x by a constant, the standard deviation won't be affected, and the mean, I'm subtracting it anyway. They're insensitive to scaling x by a positive constant. If I multiply x by 2, this changes by 2, but also the standard deviation changes by 2. Okay, so you can stretch a variable, you can shift a variable, and the correlation doesn't change. A variable with itself, by definition, this is the variance, and this will be the standard deviation squared, so the correlation would be 1. And two independent variables, by definition, this will vanish. Okay, so it's 0 for independent variables, 1 for perfectly correlated variables, minus 1 for a variable, and it's negative. Okay, so it goes between minus 1 and 1 and measures, it's a dimensionless measure of the strength of the correlation between two variables. Okay, so let's begin now and calculate, so just to summarize what we found so far. We found that if we look at log to the base 2 of size, this is the variance. And in a similar way, we found that, maybe I'll write it, it's written on the board, but I'll write it here. This allows us to find that, I'll just write it in a cleaner way, so times 1 over alpha 2 minus alpha. And we found that for the time, sigma tau squared is equal to sigma n squared times square root of 2 divided by 2 minus alpha. Okay, these are the two things we found so far. And now we're going to start talking about the correlations. And the first correlation we're going to do is between size at birth and, let's say, size at division. So what is the correlation of these two variables? In other words, I'm born small. For a given value of alpha, do I divide small or do I divide big? How are these variables correlated for a given value of alpha? Okay? Now, just as a warm-up, I can ask you about the correlation between size at birth at the nth generation and size at birth at the next generation. What is the relation between these two variables? Why? Perfect. So this variable is just this variable divided by 2. We just said that Pearson is insensitive to scaling, so these must be equal. Okay, good. Now comes a trickier thing. I want to claim what were the more natural variables for us to work with instead of VBN and VBN plus 1? Xn and Xn plus 1. And I want to claim that I can replace these by Xn and X plus 1 approximately and write this, because this will be easier for me to compute. Why do you think that's the case? What's the tool that will enable us to replace? I remind you that, maybe I used the same notation as before. I defined Xn as log to the base 2 of VBN divided by V naught. So why can I replace this variable with this variable for the purpose of the correlations? What's that? So it's not always true, and this is approximate. So what kind of approximation do we need to make here? So we know that all the stuff that we're talking about, the distribution of size, we've already used it before, is narrowly distributed around the typical newborn size, right? So I can take this function as a function of VBN, and I can, if I want, Taylor expanded around VBN. And it's not even important what I will get here. I will get here some positive coefficients times VBN. Okay, I will get here some constant plus some positive coefficient times VBN minus V naught, if I Taylor expand around the typical newborn size. So the same logic of Taylor expansion we used before justifies replacing this logarithm with a linear function in VBN. Now, if I tell you that I replace VBN by this linear function of VBN, how will that affect the Pearson correlation coefficient? No effect. Why not? Because it's a shift, constant shift, and a scale. And neither scaling by a positive constant or a shift affects the Pearson correlation. So I can do that for VBN, and I can do that in the same way for VBN plus 1. Repeat the procedure. So we've just proven that correlation of birth and division is approximately equal to correlation of birth in the next generation. And that's exactly equal, or depends if you have perfectly symmetric division or not. But for symmetric division, it's exactly equal. And this is approximately equal because of this Taylor expansion logic to the correlation between xn and xn plus 1. And now our life is going to be fairly easy because we've already had this simple relation that xn plus 1 equals 1 minus alpha xn plus noise. Remember that? That's why we chose the parallel model to begin with. Okay? And like I said, if you do a different model with size additive noise, everything, the final results we'll get now will be identical. They will not change. Okay? So now I want to calculate the correlation of xn and xn plus 1. And what I just erased told us that the way to do it is to calculate the expectation value of the products of the two variables when you subtract their means and normalize by their standard deviation. Well, what is the means in this case? 0, so nothing to subtract. So I just need to take the expectation value of 1 minus alpha xn plus noise multiplied by xn. And what is the product of the standard deviation? What did we call that? They're the same. They have the same. We used it before that they have the same standard deviation. So the product of their standard deviation is simply the variance of the log size, which we called sigma v. So this is simply sigma v squared. Okay? So now it's super easy. Why is it super easy? We get here that this is equal. Remember that unlike variance, expectation value is linear in this coefficient, so I can simply take it out. So I get two terms. I get 1 minus alpha times the expectation value of xn squared. What is that? The variance, sigma v squared. So we get sigma v squared divided by sigma v squared, so that's going to cancel. And the second term, what is the expectation value of the noise times xn? 0, because by assumption, the noise is independent. So we get that this is simply equal to 1 minus alpha. So this was one of the correlation coefficients I quoted on the presentation, that I said that cell size at birth and division, the correlation is 1 minus alpha. And already in 1980, this correlation was measured. People looked under the microscope at E. coli growing and saw that the correlation is half. Okay, so half is our first hint that alpha equals to half. So I'm saying for data, it's approximately half for E. coli. Now let's do another coefficient, kind of for the warm-up of this machinery. So the next coefficient we'd like to do is the correlation between cell size at birth and the generation time. And by the way, it's kind of the same. If I ask you what is the correlation between division and the cousin division, it's the same machinery. Now that you know how to do it, you can do any of them. And it's also very easy to simulate. Okay? So let's do the correlation of size at birth and the time. And you won't be surprised which variable, to make the calculation easy, am I going to replace with what? To make everything nice and Gaussian and simple stuff. What do we want to work with? The x. So I replace the VB with the x. Right? So I want like the xn, how is it correlated with the time of that generation? Okay? And also for that, we had a very simple formula, right? Because we saw before that the time was equal to a constant tau. That's not going to affect the correlation coefficients because we subtract the average anyway, minus alpha tau times xn. Remember we had this relation before where it was like linear in the xn with a prefactor of minus alpha? So that's what we had, plus the noise. So now, if I take the expectation value of t minus t average times xn, for xn I don't need to subtract any average. Okay, I need to do that. And I need to divide it by sigma v, which is the standard deviation of the xn, times what? Sigma t. And we calculated both of them. It's written over there. So we are kind of in a perfect position to do this calculation. And again, when we do this, which terms are, this term is not going to contribute because I subtract it anyway. Okay? Psi is uncorrelated with xn. So the only thing we get is this minus alpha tau times xn squared. What does xn squared contribute? Sigma v squared. Okay, so we get sigma v squared divided by sigma v, sigma t. So now all we have to do is plug in these formulas into this. And I'll save you this simple algebra, and you find that this correlation coefficient is equal to minus square root of alpha over 2. And this can also be measured, and you find that it's approximately, for E. coli, equals to minus a half, suggesting again a value of alpha equal to 1/2, corresponding to these other correlations. Okay, so that's the second coefficient that we wanted to do. And okay. And the last thing I want to do is discuss this collapse. Okay? So I've already told you that you can collapse the size. I showed you this collapse of the distribution. So now we're in a position to understand it. If I look at the distribution of cell size. Okay, let me just find it. Okay, so if I look at the distribution of cell size, I know that this is the variance of log to the base 2 of cell size. But now I want to look at the let's say I want to look at the coefficient of variation of the size distribution. So again, coefficient of variation is the standard deviation divided by the mean. It's a dimensionless measure of how broad the distribution is. So I can do that for the time distribution, and I can do that for the size distribution. Okay? And if I do that for the size distribution, I know that x equals to log to the base 2 of v over v naught. So I can say that my size is simply 2 to the power of x, or e to the power of log 2 times x. Okay? Up to a v naught. So maybe it's like v naught e to the log 2 times x. Okay? So if I want to look at the coefficient of variation of this thing, for the coefficient of variation, I can drop this V naught. It's just a prefactor. It doesn't affect the width of the distribution. So let's look at this guy, e to the log 2 times x. X is a narrow distributed variable, right? We know that the variance is small. So the mean of this thing is 1. And the variance, I can Taylor expand and find that this is approximately 1 plus x log 2. Okay? So I can say that the coefficient of variation, we call it CV, the CV is approximately equal to the variance of x times log 2 squared divided by 1, because the mean is about 1. Sorry, I made a mistake. That's for the variance. So the CV, I need to take the standard deviation. So the square root of this. Everybody on board? No? Should I repeat? Yes? OK. So coefficient of variation is defined as standard deviation divided by the mean. It tells me whether this distribution is narrow or wide, right? It tells you how wide it is compared to the mean. I'm saying that size is up to a constant, e to the log 2 times x, where x is the variable we worked with so far, because it was log to the base 2 of size. If I Taylor expand this, I get 1 plus x log 2. The variance of this thing is the variance of x times log 2 squared. So the standard deviation is the square root of that. And to get the coefficient of variation, I need to divide by the mean. But the mean of this thing is approximately 1, because x has a negligible mean. Okay? So I find that the coefficient of variation of the size distribution is basically this thing. And we found what it is. This is simply log 2 times sigma v. Okay? And similarly, so this is CV of size. And I can also calculate CV of time. What is the CV of the time distribution? Well, I know the standard deviation of the time distribution is written right there. It's sigma n, okay, times square root. Sorry, this shouldn't be here. It's like this. Sigma n times square root of 2 divided by 2 minus alpha. All of that divided by the average, which is tau. Okay? Now I can plug in CV, and I get log 2 times sigma n over tau, 1 over square root of alpha times 2 minus alpha. OK. We already realized, so now if I take the ratio of these CVs, I can take the CV of time divided, or let's take time. Let's take size. Let's take CV of size divided by CV of time. So I need to divide this by this. Sigma n over tau is common to both, so it drops. And I get a super simple prediction that this is equal to log 2. Okay. Times 1 over times square root of 2 times alpha. Divided by square root of 2 times alpha. Okay, because the square root of 2 minus alpha drops, and we just have the 2 and the alpha. What do we predict? We predict that for alpha equals to half, if we have this alpha equals to half, the size distribution is narrower than the time distribution, but by about 30%, by a factor of log 2. Okay? So that's a prediction, and that's exactly what the experiments give you, okay, or do a good approximation. And this is essentially the reason for this collapse that I showed you earlier experimentally. Because the size distribution is narrower than the time distribution by a factor of log 2, simple algebra will tell you that log to the base 2 of size should collapse with a normalized time distribution if you shift them both by, the size distribution is naturally centered around 0, and if you divide by the average time and center by subtracting 1, you will find that these two are predicted to be Gaussian and to collapse. Okay? So what did we see? We saw that the model captures well the correlations and captures well the distributions associated with the statistics of the data. I want to show you another way of seeing this, these correlations, okay, that will become useful for us tomorrow as well. So what you see here in this cloud are single E. coli cells. So the x-axis is the size at birth, and the y-axis is the size at division. So you get this cloud of points. Most of them are here, right? Also correlation coefficients. You can only compute correlation coefficients when you have variability in the data. If E. coli was absolutely perfect, all the data would be 1 point here, and you wouldn't be able to measure correlation coefficients, measure distributions, or do anything. So all of what we're doing now is possible because of the natural variability between cells. Again, similar in spirit to Luria-Delbruck. Okay? So the cells are pretty good. This is a heat map, and most of the cells are sitting here. But these are outliers that were born small or born large, and then they also divide small and divide large. And by now you understand that this birth and divisions are going to be correlated. Okay? So one way to understand these correlations is by doing the Pearson correlation coefficients that we just computed. Another way to do it, similar but slightly different, is a procedure called binning. What is binning? I take all the cells in a given rectangle here for a given size at birth. I approximately fix the size at birth, and I average over all these cells here. Okay? Now, within the model, size at division is a function of size at birth plus some noise. Okay? Again, the details of the noise are not super important. So by binning, I essentially get rid of this noise in the Langevin-like equation we wrote. And therefore, I extract the underlying relation between size at division and size at birth. Okay? So this is real data for E. coli. And if you do this binning procedure, you reveal this dependence that's hidden in this data, where all these red circles, they're not theory. They're just the binned raw data. And now you see they sit on this nice straight line. Okay? And you find that this line has a slope very close to 1. What does that tell you? It tells you that size at division is equal to size at birth times approximately 1. That's the slope plus a constant. So that's, again, this equation for this adder, where you're adding a constant volume from birth to division. So that's another way of seeing this, getting at this value of alpha equals to half corresponding to these adder correlations. Is that clear? Okay. So again, this is the same. You can do the same procedure of binning for the size at birth and the time. And in this case, if you bin, you get a nonlinear relation. So the red circles are the bin data. And the black line are the prediction. And the prediction is very simple. What is the functional dependence we expect between birth and division time? Well, cells grow exponentially. So I can write that VB e to the lambda t should be equal to the size at division, which is, again, if I suppress the noise by binning, is equal to VB plus a constant. This equation defines time. It will tell me that time is the logarithm of 1 plus delta over VB, this function that's written here. And that's the black line here. Okay, so that's another way, alternative way, instead of calculating the correlation coefficients, calculating this relation between the variables upon binning. Okay, so you can do it for any variables you like, and so on and so forth. I need to finish, but I'll come back to this in a second. So I want to say, at the beginning of the course, we had this quote, what is true for E. coli is true for the elephant. Okay? So we can ask, does this work for other microbes? And the answer is yes. So together with our collaborators, we showed that budding yeast, remember I showed you in the very first class these videos of E. coli, budding yeast, and archaea, right? So all of these also follow this slope of 1 corresponding to these other correlations. And so by now, there are many species of bacteria that were shown to follow these other correlations. And people have also shown it from mammalian cells. And like I mentioned, these archaea that grow at very high salinity concentration. So they're quite a broad class of organisms for which these other correlations of, and size control is followed. This leads to a natural question related to the first part of the course. Why did this evolve? Is this size control strategy optimizing some sort of fitness at the population level? And we have a lot of works on that, and so far we couldn't find a convincing model where this happens. So, somebody told me yesterday that it seems like all the questions have been answered, and the truth is very, very far from this. Like I'm giving you the tip of the iceberg, and for everything I'm presenting, there are a zillion open questions. And one open question is, why have adders evolved across the tree of life? You saw on this slide that these are very distant organisms from all over this phylogenetic tree of life, and we don't know what's good about this adder size control that they have evolved. And as a kind of last point for today, well, I'll go a little bit over time, and I want to make 2 more points today. So one point is that the model can also fail. Okay, so the model for any value of alpha is not able to explain the statistics of these mammalian cells, lymphoblasts. Okay, so what was done in Nathalie Balaban's group, she measured the correlation of the timing between cousin cells. Now, within the models we discussed today, within the stochastic models we discussed today, you can convince yourself, either analytically or with simulation, that if I take a cell and its cousin cell, the correlation between, say, the timing should be the correlation of the mother and daughter squared times the sister-sister correlations, right? Because that's kind of how you get from here to here. But that means, in particular, that cousins should be always less correlated than mothers and daughters. And what she found was that cousins were significantly more correlated than mothers and daughters. And that's beyond the stochastic model we presented here. And they attributed this to the existence of circadian clocks that make cells of the same generations more similar to each other. Okay, so again, I'm happy to talk more about it offline, but I'm saying this is an example where this stochastic model, not just for the adders, but for any value of alpha, is not able to capture these statistics. And that's okay, because it tells you, okay, we're missing an ingredient, and in this case, they postulated that this ingredient is the existence of circadian clocks. The last thing I wanted to tell you, which I think is, I actually prepared the calculation, and maybe I'll go over it tomorrow if we have time, but when you look at this single cell statistics, this is this heat map, we said that the binning gives you a slope of 1, right? And indeed it does. This is the red bin data, has a slope very, very close to 1. And about a year ago, when I was preparing an ERC presentation, I showed it to one of the physicists at Weizmann, and he was telling me, I don't believe what you're telling me, because when I look at this cloud of points, the contours, you can kind of look by eye and see that this cloud has a structure that seems shifted compared to this bin data of 45 degrees, right? So he's saying, so I don't believe this binning, because by eye it looks like everything is really pointing more in this direction, right? This is this elliptical contours that you find. So it turns out that this is both true and a natural prediction of the model. So I don't need to change the model at all. We didn't publish this, but we're working on it. And if you take the very same model that I thought, I understand everything about it, and it turns out that if you calculate the contours of this heat map of VB and VD, first of all, they are approximately elliptical. And the angle of this ellipse, for the case of the other model, the alpha equals to half, you can compute, again, I can show you the calculation tomorrow if you like. The angle turns out to be arctangent of 3 plus square root of 13 over 2. This comes out, which is approximately 73 degrees. So the prediction of the model is that it leads to contours that have an angle of 73 degrees, but if you bin, the binning will give you this 45 degrees corresponding to this other. So it looks simple, but it's quite subtle, and I think there is another kind of lesson here for handling data of statistics, because what I told you today is specific to size control, but a lot of the lessons of dealing with data and with variables and with statistics are completely general. Pearson correlation coefficients, binning, and contours. Contours are essentially equivalent to doing principal component analysis, which is done all the time. So the take-home message is that if you're analyzing your data using PCA or binning or correlation coefficients, make sure you can validate the result within a null model, right? Like here we have a simple model that can tell me, okay, I make this and this prediction for the contours, this and this prediction for the binning, I know what to expect, and now I can compare it to the data. Because sometimes people just take the data, do the PCA, or do the binning, and draw a conclusion without having a model at the back of their mind. And that's problematic, because you're kind of jumping over, and you might get to the wrong interpretation. So we also have works on that and on the subtleties of binning, and how binning can give you, without the appropriate null model, artifacts. So if you're interested, I can refer you to these papers. Okay? So sorry for going over time. Tomorrow we'll show that even though the model is very successful at capturing all the statistics of the data, it misses very important biological features, namely the coupling to DNA replication. So that will be the focus of tomorrow. And please pass back the signature sheet.